my dear cell champions. I believe everyone who's out there is doing perfectly great, and today I am here with a gift to you all. That is the passing package for biology, and this is for PU1 students. So, what does this particular video contain? Why should you watch this? First thing, I am not planning to make it very long. Second thing is that you will get all your important questions that might come in your exam over here. Now, few of it I might be explaining. Okay, a few of it I might be just saying that these are the set of questions. You can take a screenshot or you can ask for the PDF also. You will be getting it in the WhatsApp group, but if you do not find it, you can just put down the comment in the comment section. Now, there might be certain questions that you feel that you are not able to understand by your own, even if I give you the questions. Then you can comment that also down. I will be taking a separate video where I will be explaining all those important concepts to you. Okay, will that work? I hope that works. So let's get in, and yes. So I have segregated the chapters according to its weightage, which was there in the previous year. So this year there is no blueprint that has been given. So according to the previous year blueprint, cell the unit of life carried 12 marks, my dear children. So here are certain 2-marker questions which you should definitely not miss. So state the cell theory. So over here you have to talk about Schleiden and Schwann. What did they do? Basically they said that all living organisms are made up of what, my dear children? Cells. So you should be knowing about that. Okay, and there was one more guy who added one more thing. Okay, that is Rudolf Virchow. He said that cells which are there, they do not come from the sky, they come from the pre-existing cell. The cells which were there, they divide, and that's how you get new cells. So this is what you have to talk about cell theory. You can write Schwann and Schleiden and also Rudolf Virchow over here. Now, what are mesosomes? Mention any one function. Now, mesosomes are found in prokaryotes, my dear children. And how are they formed? So they are the extensions of the plasma membrane. You know plasma membrane? So it goes inside, and this structure that is formed is known as mesosome. But what is the use of it and how it is formed? So basically, these extensions are in the form of vesicles, okay, tubules and also lamellae. They help in cell wall formation, that is the use. They help in DNA replication and the distribution to daughter cells as well. So how do they do that? Basically, they have so many enzymes over them, which is why it helps in cell wall formation, DNA replication, and do this and that. So they also help in respiration, secretion processes to increase the surface area of the plasma membrane and the enzymatic content. So all these things are from your NCERT. I'm just here to tell you what you should be including in your answer. So these points you have to include. You're asked about any one function, so you can put any function. Okay, but remember they have so many enzymes, and since prokaryotes do not have membrane-bound organelles to do different functions, that is why they have mesosome. So in the case of eukaryotes, you have mitochondria for respiration. Over here you will be having mesosome with all the enzymes needed for what? Respiration, my dear children. Now list the type of ribosomes found in prokaryotic and eukaryotic cell. So prokaryotes means what? They are primitive cells. They do not have membrane-bound organelles. They do not have a nucleus. Okay, the genetic material is just put there. Okay. And this region where the genetic material is there, which is not lined by any membrane, you call this region as nucleoid, my dear children. Whereas in the case of eukaryotes, you have the nuclear membrane, nucleus, and everything over there. Now here the ribosomes which are there, they are quite different. How they are different, my dear children? The eukaryotic ribosomes are ATS, that is the unit, sedimentation coefficient. Okay, Swedberg's unit. Okay, while the prokaryotic ribosomes are 70S. Okay, eukaryotic 80, it will be more, okay, and ribosomes of prokaryotes are 70S. Now again, each ribosome has 2 subunits in the case of eukaryotes and obviously prokaryotes. The larger subunit will be there and there will be a smaller subunit. Over here, when you have to divide 80, it's not like 50-30, always remember, it is 60-40. So 60 and 40 is equals to 100, no ma'am. So basically what does this suggest? It is a sedimentation coefficient. Okay, so the largest subunit basically takes probably sediments in this particular range. In this particular range where it is what? 60. This is sedimentation rate. Whereas the smaller subunit might be sedimenting like this over this particular range. Okay, now when you put both of them together, they might be sedimenting around what? 80, which is why it is ATS. Okay, and this was Swedberg's unit, my dear children. Okay, now if we talk about 70S ribosomes of prokaryotes, they can be divided into 50S, that is larger subunits, and 30S, that is smaller subunit. So these are the type of ribosomes found in the prokaryotes and eukaryotes. You should be knowing about them. Going ahead, difference between RER and SER. What do you mean by RER? Rough endoplasmic reticulum. SER, smooth endoplasmic reticulum. The structure, why do they appear rough? Because they have, my dear children, what? Ribosomes on its surface. Okay, making it appear rough. What about smooth endoplasmic reticulum? They do not have ribosome on its surface, which is why they are smooth. Now function, what does RER do? Now whenever you have ribosomes, please remember ribosomes major function is to produce protein. So if rough endoplasmic reticulum has ribosomes, that means this guy will be doing what? Protein synthesis. What about SER? SER will be responsible for lipid, that is fat detoxification, and also for calcium storage. So in our muscles, the smooth endoplasmic reticulum has a modified version, which is known as sarcoplasmic reticulum. So there it stores calcium. There it stores a lot of calcium, because for muscular contraction and relaxation, you need lots of calcium. So this is acting as a storage unit as well over there. Okay. Now name the 4 endomembrane organelles. The 4 endomembrane organelles are endoplasmic reticulum, my dear children, Golgi complex, lysosome and vacuoles. Why they are known as endomembrane organelles? Because their functions are kind of coordinated. From endoplasmic reticulum, things will be going to Golgi apparatus for modification. From Golgi apparatus, you will be getting lysosome, you will also getting vacuoles and things like that. So since their function is being coordinated, you call the 4 endomembrane organelles to be them. Cool. Now functions of Golgi apparatus, SER, lysosome, vacuole, chloroplast and mitochondria. All of the organelles you should be knowing. So Golgi apparatus, it modifies, stores and packages proteins and the lipids. So from RER and SER you get protein as well as lipids. So the modification is done by Golgi apparatus. Smooth endoplasmic reticulum again synthesizes lipids and detoxifies harmful substances. Okay. Lysosome, it has hydrolytic enzymes, my dear children, which is responsible for killing any foreign particle that comes through. Okay, it has digestive enzyme or hydrolytic enzyme. So break down waste materials and cellular debris using the digestive enzyme. Now they are known as a suicide bag of the cell, because once they are released, probably they are not worried if the cell is being killed, but they are anyway going to kill the foreign particles. Now vacuole, obviously they store nutrients, water, waste. In the case of plants, they store certain pigments also. They provide structural support in the case of plant. If you see a plant, almost 90% of the plant is being utilized by the vacuole itself. Okay, chloroplast, it's a plastid. And this chloroplast, the plastid which is there, chloroplast, it has high amount of chlorophyll pigments, my dear children. So it helps in photosynthesis to produce what? Glucose and oxygen. What about mitochondria? It is the store, it is basically the one that will generate what? ATP. Okay, it will generate the ATP through the process of cellular respiration. So you should be knowing about each of the function in detail as well, but just giving you a gist over here. So yes. Now what is active transport? Give an example. Now whenever we say active transport, that means over here you are spending money for transporting something. So over here in the case of plants and animals, in our body there is no money exchange, right? So what is money over here? It is ATP, my dear children, adenosine triphosphate. So transport in which ATP is being utilized, this is the money that is being utilized, is known as active transport. For example, sodium and potassium pump, which is right over there. So these are the 2 marker questions that you should definitely go through once before going to the exam from this chapter. Now apart from that, there are 3 marker questions also. So list out the functions of plasma membrane. You should be knowing the functions of plasma membrane. What are those? Regulates the entry and exit of the substances, provides what? Mechanical support. Okay, and then on the plasma membrane there might be receptors, so this might be helping in cell to cell signaling. Okay, then helps in what? Cell communication through the receptors, everything. So you have to write all the points right over here. Now classify the type of plastids based on the type of pigments which are there. So you have chloroplast, chromoplast, leucoplast. Chloroplast contain chlorophyll pigments which helps in photosynthesis. Chromo as in colorful, so it contains carotenoids. Okay, and it gives colors to what? Fruits and the flowers and everything. Okay, it's not just carotenoids, it also contains xanthophylls, the brown pigment, and the carotenoid is the red and orange pigment, xanthophylls is the yellow and brown pigment. So all this pigments are there in the chromoplast, and this is the one that gives the flowers and fruits there beautiful colors. Okay, now leucoplast, it is there for storage. It is exclusively there for storage. What do they store? If they store carbohydrate, my dear children, they are known as amyloplast. If they are storing proteins, they are known as eleuroplast. If they are storing fats, they are known as elioplast. So elio I remember by oil. Elio has some oil in it, O-I-L, oil. So oil is a fat, so basically fats, elioplast. Okay, either way you can remember, but yes, remember it. Now cell wall is non-living rigid structure made of 3 layers. Mention the wall layers and its chemical components of bacteria. So now 3 layers of bacterial cell wall and chemical components. First is glycocalyx, my dear children. Okay, so glycocalyx, it is basically, it can be in the form of capsule or it can be in the form of slime layer as well. It is made up of polysaccharide. Now inside that you have the cell wall which is made up of what? Peptidoglycan. Now you have plasma membrane. Plasma membrane is made up of what? Phospholipid bilayer that you have. So basically it's not like mention the wall layers. Yeah, mention the wall layers. Yes. So these are the three walls that you have: glycocalyx, cell wall and plasma membrane. Please remember, what is it made up of? Glycocalyx is made up of polysaccharides, my dear children, and can be in the form of capsule or slime layer. Cell wall is made up of what? Peptidoglycan in the case of bacteria. Everyone has different things. Fungi has a cell wall made up of chitin. We have, not me, plants have cell wall made up of cellulose. Okay? Yes. Now plasma membrane, phospholipid bilayer, obviously it's common for all. Now going ahead. Name the three components of bacterial flagellum. So bacteria has some flagella to move around, right? So what is it made up of? It has filament, long helical structure for movement. There is a hook that will connect the filament to one part of the body, that is basal body, my dear children. And what does this do? It anchors the flagellum to the cell membrane. It attaches it to the cell membrane. So filament, hook and basal body, which is there. Now what is cytoskeleton? Write any two functions of cytoskeleton and its function. So basically cytoskeleton is a network of protein filaments which is there. Okay, it provides the structure. It provides the structure and the mechanical support. So how do you write the function? Maintains the cell shape and provides mechanical support, and aids in intracellular transport and cell division as well. So during intracellular transport, when things are being transported in the cell, and also while cell division is happening, and also for mechanical support, for providing the cell a proper shape, all you need is what? Cytoskeleton, my dear children. Now which organelle is called packaging apparatus and why? Packaging apparatus, basically what happens? From RER and SER you get modified things, you get things. Now in the Golgi apparatus it gets modified, and this particular Golgi apparatus is the one that will package it and will be sending it out of the cell or probably within the cell or whatever it wants. Okay, so packaging organelle, and the reason is organelle is what? Golgi apparatus, and the reason is that it modifies, sorts and packages proteins and lipids for secretion or for transport. Cool? Chalo. Now, difference between prokaryotes and eukaryotes. Nucleus, I told you, they do not have a true nucleus, my dear children. Their genetic material is present in a nucleoid region, whereas eukaryotes have a well-defined membrane-bound nucleus. What about organelles? Prokaryotes lack membrane-bound organelles like mitochondria, Golgi apparatus and endoplasmic reticulum, while eukaryotes contain these organelles. So, the single membranous organelle, for example, ribosome is there in both prokaryotes and eukaryotes. The double membranous organelles such as this are not there. Okay, now talking about the cell size, prokaryotic cells are generally smaller, 1 to 10 nanometer, compared to eukaryotic cells which are larger, 10 to 100 nanometer. Now DNA structure in prokaryotes, DNA is circular, my dear children, you might have seen, okay, and whereas in eukaryotes, DNA is basically linear. It's a linear structure. And this linear structure is compact. It is compacted with the help of histone proteins which are there. Whereas in the case of prokaryotes, you don't find that. Okay. Now, diagram, mitochondria and chloroplast is important. Please practice the diagram and go for the exam. This is a 3-marker question that can be coming from here. Cool. Going ahead. So, you see the time that we are taking is almost 12 minutes by discussing one chapter. So, obviously, we cannot afford to discuss all the chapters, which is why I have put the questions like this. Please don't worry. You can take a screenshot or ask for the PDF as I said. But you will be getting it, you can just go through. So these are the questions. Again, take the screenshot if you want. This is 5-marker questions. So prepare the diagram of plants. Now this is a diagram based question, my dear children. Again, diagram based question. So that question can come for 5 mark also. Okay. Explain the fluid mosaic model. Explain the structure of nucleus with a neat label. Again, there is a diagram question. So D. You see the diagrams. Okay. These diagrams are really, really important. The difference between plant cell and animal cell. Describe the structure of prokaryotic cell. Identification of organelle with structure with the functions. We have already done the organelle thing, but still you have a lot to do. Now name the following powerhouse of the cell, that is mitochondria, site of steroidal hormone synthesis, reservoir of hydrolytic enzymes, packaging units of the cell, locomotory structures of the cell. So all these things are really, really important. You should know the function of all the organelles, otherwise you will not be able to answer it. For example, randomly I am taking reservoir of hydrolytic enzyme. I just told you it's lysosome. Packaging unit of cell, Golgi apparatus. So if you do 2-marker, 3-marker question properly, you get 5-marker. But you should not be focusing like that. You should be focusing on 5-marker question. This can give you 2-marker and 3-marker. That's how it should be working. So these are the questions from this chapter that you should definitely not miss. Okay. Going ahead, animal kingdom, it's a 9-marker chapter. In the previous year it was like that. So in that also you have 2-marker and things like that. So first question is, what is bioluminescence? Give an example. Bioluminescence, you know, you might have seen people run to see bioluminescent things over there. So it's an ability of an organism to produce what? Light at darkness, and they look so pretty. Example is what? Fireflies. You have certain jellyfish and some deep sea fishes like anglefish. Okay, now anglefish. Now you have all vertebrates are chordates, but all chordates are not vertebrates. Explain, justify. So basically, my dear children, if you talk about chordata, phylum Chordata, you have three subcategories. There is Urochordata, there is Cephalochordata, and there is Vertebrata. On what basis are they categorized? How did they come under chordata? Because all of them had notochord any time in their lifetime. In the case of Urochordata, they have the notochord in the larval stage in the tail. Okay. In the case of Cephalochordata, they have from head to tail throughout their life. In the case of Vertebrata, they had the notochord, but this is replaced by vertebral column. So I can say that all vertebrates are chordates, but I cannot say that all chordates are vertebrates. Okay, so all vertebrates are chordate, but all chordates are not vertebrate. Justify, because chordates have a notochord at some stage of the life, but not all develop a vertebral column, my dear children. Obviously, not all of them are developing a vertebral column, right? Vertebrates is a subgroup of chordates where the notochord is replaced by vertebral column. Example, Amphiosis, which is there, is a chordate, but it is not a vertebrate, my dear children. Not just for you to understand this thing. Now don't go right, Amphiosis is a vertebrate but not a chordate. It is a chordate but not a vertebrate. Okay. Now differentiate between coelomate and acoelomates. So you know we have three germ layers. Okay, ectoderm, mesoderm and endoderm. The mesoderm is the middle layer over there. Now in the mesoderm, if you have a cavity over here, suppose this is the mesoderm. So this cavity is being lined by the mesoderm, my dear children. You call this as coelom. So animals with true coelom are the one that is known as coelomates. Okay. Here the coelom is being covered by the mesoderm which is there. Example, annelids, mollusks, chordates, etc. What about acoelomates? The one that does not have the coelom at all. The body cavity is itself not there. So animals without a coelom, body cavity is absent. They are known as what say acoelomates, and the example is what? Platyhelminthes. Okay, going ahead. Difference between radial and bilateral symmetry. What do you mean by radial symmetry? For example, I have an organism, I can cut it in through different planes, but still I will get equal halves. So this is known as radial symmetry. I say that this organism can exhibit radial symmetry. Example, you have cnidarians, my dear children, like jellyfish and hydra, whatever you cut, you will get the same equal halves. Bilateral symmetry, like us, for example, you cut it from between, that's how you get two halves, equal halves. Otherwise, you are not going to get two equal halves. So bilateral symmetry is when body can be divided into two equal halves along one plane only. Example, chordates, arthropods and annelida. For example, think of a crab. Crab you can cut only from the middle, giving you two. You also say annelids, for example, any worm, cut into two, you will get two. But you cannot cut through multiple planes. You think of it and then you remember. Okay, now what is metamerism? Name an organism exhibiting this particular character. Metamerism is basically when there is a segmentation externally as well as internally. Okay, there is a serial repetition of these segments. It is seen in annelids, that is earthworm. Now difference between external and internal fertilization. When the sperm or basically the male gamete and the female gamete is fusing outside the body. Okay, outside the body, this is known as external fertilization. For example, in the case of frogs, fishes and everyone, they have external fertilization. What about internal fertilization, which happens in human beings? Over here, the sperm as well as the gamete, the female gamete which is there, it is fusing inside the body of an organism. So this is internal fertilization, my dear children. Example, mammals, birds and reptiles. Cool. Now name the animals exhibiting the given characters. Organism with a sucking and a circular mouth. This is a very important question. Lamprey, my dear children, it's an agnathan. Does not have jaw, which is why its mouth looks like this. Okay, it's a sucking mouth and a circular mouth that they have. Now organism in which notochord is present only in the larval stage. It is what? Urochordata, that is ascidian, my dear children. Going ahead. Okay. Difference between direct and indirect development. So direct development as in, for example, the young ones which are being produced, it resembles the adult ones. Okay, there is no larval stage included. For example, humans and birds are like that. What about indirect development? Whether there is a larval stage, pupa, this, that, things like that. So young ones pass through a larval stage before becoming an adult. This is known as indirect development. Now mention any two functions of flame cells. Now flame cells, my dear children, they are very much important for excretion and also for osmoregulation, found in platyhelminthes. Okay, so flame cells helps in the removal of the nitrogenous waste like ammonia, and they maintain water and ionic balance in the freshwater flatworms like planaria. Flatworms, basically you're talking about platyhelminthes. Okay, now give any two parasitic adaptation of flatworms. Parasitic as in because they are going to live inside a host or outside the host, whatever they do, basically they are going to damage or basically cause problem to the host. So they are known as a parasite. Okay, so they have hooks and suckers if they are living inside. So found in tapeworms, my dear children, for firm attachment to the host intestine. So tapeworms, they stay attached to the host intestine with the help of the hook and the suckers. Now they have high reproductive capacity. Why? Because suppose our defense system is really strong, we kill certain tapeworm, but still, suppose there are so many tapeworms being produced, there are high chances that they could survive. So produces a large number of eggs to ensure survival despite host defenses. Okay, so you have to write this and come. Now, these are the 5-mark questions that you should not be missing. List 2 classes of chordates which are oviparous and development is direct. Characteristics of mammals as well you have to take. Now, assign the following animals to their respective phyla: Planaria, Gorgonia, Hirudinaria, Cucumaria, Ascidia. Okay, assign the files organs of, oh my god, wait. Assign the organs basically, that is what? Malpighian tubules, radula, parapodia, comb plates, proboscis, etc. So you should be knowing about all this thing really well. Okay, so for example, proboscis found in hemichordata, comb plates found in ctenophora and things like that. So you should be knowing Malpighian tubules found in amphibian. Malpighian tubules found in arthropoda. Okay, so you should be knowing about all this thing. So go through the phylum properly, then only you would be able to answer. Define the following terms. I told you bioluminescence, metamerism, poikilotherms. What do you mean by poikilotherms? Those who do not maintain a constant body temperature. Okay, those that are not maintaining a constant body temperature. For example, you talk about reptiles, you talk about amphibians, you talk about, yeah. So basically they do not maintain, even fishes, okay, they do not maintain a constant body temperature, which is why the energy requirement will be really less. Pseudocoelomates, where there is a false coelom, my dear children, bilateral symmetry we also study. Differentiate between chordates and non-chordates. You should be knowing differentiate between chondrichthyes, that is cartilaginous fishes, and osteichthyes, that is bony fishes. General characteristics of arthropoda and echinodermata. Again, anything can come. No, it can be annelida and that also. But yes, these are the probable things for 5 marker. Cool. Going ahead. Oh my god, what's in there? So my dear children, if you are someone who is doing PU1 but still you are planning for KCET, you have already started off your preparation full swing. Okay, so here we have the Abhyas KCET test series. I know it might not be totally relevant to you, but if you have someone in 12th grade as well who is preparing for KCET, you can suggest them also. So we are launching Abhyas KCET test series where we are providing 35 full syllabus as well as chapter wise tests, my dear children. Detailed video solution and analysis. There will be chapter wise one-shot videos, and also paper analysis has been done for the past 5 years. So this is going to be really crucial and really important for someone who is preparing for KCET. So if you think that we can also try it out, you can try it out. You can start from this particular year itself. So please do that. This is for 2025 and the offer price is 1499, where you will be also getting PU2 test series for free. Okay, so as I said, might not feel relevant to you, but it will be really important for anyone who is in PU2 or anyone who is aspiring to, even in being in 11th grade, you are aspiring to crack KCET, it will be really helpful for you. Yeah? Chalo. Now, plant growth and development, this is the next chapter that we have. According to previous year, 8 marks was there. Now, 2-mark question we will discuss. ABA is also known as stress hormone, my dear children. Give reason. So ABA is known as abscisic acid. Why is it known as stress hormone? Because it allows the plant to survive during stress conditions. For example, there is too much amount of salt content in the soil. There is too less water in the soil. So, or there is high temperature in the soil. At this point of time, abscisic acid will protect the plant. It will help the plant survive, which is why it is known as stress hormone. It is being released when plant is under stress. So how does it manage it? Basically, for example, there is less water in the plant's body. It will close all the stomata so that no water gets lost. Okay. Or probably there is a seed, and this seed cannot germinate as of now because of lots of problems. So it will induce dormancy. It won't allow the seed germination to take place over there. So this is the function of ABA. Now differentiate between dedifferentiation and redifferentiation. When I say, for example, a cell has a function now. Okay, it has a function now. I say that this is differentiated. I say that this is differentiated. It has a proper function now. Now suppose it loses its, it gains its ability to divide. when it had a function, it was losing its ability to divide. It was just doing the function. It was mature. Now it has again regained the ability to divide. It will continue division. This is known as de-differentiation. Ulta of differentiation, okay, opposite of differentiation. This is known as de-differentiation. Now again it thought that I have to be mature. I cannot go like this. So basically again, when it again loses its ability to divide, my dear children, you call this state as re-differentiation. Okay, so de-differentiation is where the mature cells regain the ability. We are talking about this, okay, de-differentiation. It regains the ability to divide. Example, formation of cambium from the parenchyma. So from parenchyma cells, again, they get de-differentiated. That's how they form the cambium. Now over here, re-differentiation, de-differentiated cells, these cells, okay, they develop into specialized cell. They are now not going to divide. They have lost the ability to divide. This is known as re-differentiation. Just a second, okay. Now we have defined plasticity and you have to give an example. So for that, what do? So plants follow different pathways in response to environment or phases of life to form different kinds of structures. This ability is known as plasticity. Basically, it's like a plastic. You can mold it in different form and just use it. So in the case of plants also, in the juvenile stage, they might have different form to survive in that particular condition, and when they grow old, they might have different shape to survive in that particular condition. This is known as plasticity, and this is very important. Now just give me a second. Okay, so yes, we spoke about that, and now we go ahead. So heterophyly is an example of plasticity. The same thing has a different subgroup, that is heterophyly, my dear children. What do you mean by that? It refers to different leaf forms in the same plant due to genetic, environmental or developmental factors. Over here the leaves are getting different, okay, in the same plant, my dear children. Okay, why it is happening? Due to genetic, environmental or developmental factors. So buttercup, if you see, there's an example that is given in your book. Difference of shape of leaves produced in air and those produced in water. So I'll just show you the difference. This is the leaf that is there in the terrestrial habitat. This is the one that is there in the water. So this is an example of heterophyly. Okay, it's not like they are juvenile or mature. No, it's not like that. Over here I'm talking about different environment are there, which is why they have different leaf forms. Okay, that is example of heterophyly. Now what are plant growth regulators? Mention the types with an example. So plant growth regulators, they are chemical substances, my dear children. If you have seen the PU1 model paper, and there I have solved this question. Okay, so they control the plant growth. You have two, growth promoters as well as growth inhibitors. Growth promoters as in the one that will help in the growth of the plant. For example, auxin, gibberellin, cytokinin. I'll tell the function of each. One function that you have to definitely remember. Auxin is responsible for cell elongation. So if all the cells are elongating, obviously it will lead to plant growth. Now gibberellin is responsible for inducing germination. It says that you have to germinate. G for germination, G for gibberellin. Cytokinin, the name suggests it is responsible for cell division. Cytokinin, cell division. So cells divide, obviously the plant will grow. Now growth inhibitors, you have abscisic acid and ethylene, my dear children. Abscisic acid is basically stress hormone, I told you, and ethylene is a gaseous hormone which is responsible for fruit ripening. So if we keep a ripened banana with other bananas, it will get ripe. Why? Because of the gaseous ethylene which is being released. Now give an example each for natural as well as synthetic auxins. For natural auxin, you have indole 3-acetic acid. Okay, you have synthetic auxin, my dear children, that is 2,4-dichlorophenoliacetic acid, 2,4-D you call it. Auxins like IAA and indole butyric acid, IBA, also are natural. Okay, so both IAA and IBA are natural auxins, my dear children, which are isolated from the plants itself, whereas naphthalene acetic acid, that is NAA, and 2,4-D that you see over here, both are synthetic auxins, okay, chemically made. Now name the gaseous PGR, that is plant growth regulator. Mention its application. I told you gaseous PGR is only one, that is ethylene. It is the only gaseous PGR. Application is used for fruit ripening and the flower induction in the pineapples. Okay, going ahead. Yes. So what is respiratory climacteric, and name the growth hormone responsible for it. So what happens during fruit ripening? The ethylene hormone, it makes sure that the respiration rate is way too much. Why, my dear children? Basically fruit ripening as in you have to do a lot of work. The cell wall has to be softened down, the starch has to be broken down. So there are so many enzymatic activity, and for all these enzymatic activities you need lots and lots of ATP. And where do you get ATP from? From the process of respiration. So obviously for fruit, when fruit ripening happens, the ethylene will make sure that the respiration rate is way too much so that it gets enough amount of ATP for the ripening. Get it? So respiratory climacteric is a sharp rise in respiration rate during the fruit ripening. Because enzymes like amylases, pectinases and cellulases break down starch, soften cell wall and convert complex sugar into simpler sugar which needs energy. Hence the rate of respiration is what? Increased. Now ethylene is responsible for this particular process. Over here, okay, so the picture has come over there. So graphically represent the ideal sigmoid curve. Sigmoid curve, you will see a lag phase, then there is an exponential phase, then there will be a stationary phase because all the resources might be ending out. So that is why there is a stationary phase as well. So time and size, you have to draw this. Please understand this particular graph and then only go for exam. Both this and the J-shaped curve as well. Cool, so this is a 2-marker question. Talking about 3-marker question, what do you have? Briefly explain the physiological effects of auxin, gibberellic acid and cytokinin, ethylene and abscisic acid. So over here, my dear children, auxin promotes cell elongation, apical dominance. Apical dominance as in basically it says that, hey, plant, you have to basically grow like this, not laterally. So that is apical dominance. Root initiation and fruit development is also done by auxin. Gibberellin induces stem elongation, seed germination, very important, and fruit setting. Cytokinin helps in cell division, my dear children. It delays senescence, old age, or what's the aging of the plants. It is delayed by cytokinin. What about ethylene? Ethylene promotes fruit ripening, senescence. Opposite of it is done by cytokinin. Okay, it promotes senescence, it delays senescence. Now abscisic acid, it induces dormancy, closes the stomata under water stress and inhibit the growth. Now, what are the characteristics of PGR? They are organic. So many questions coming, right? They are organic substances produced in plants, regulate growth, development and responses to the stimuli, my dear children. Whatever stimulus is there, depending on it, the hormones will be released. Now, actin, very low concentration, can be growth promoters or growth inhibitors as well. Remember, they are organic things. They help in growth, development and responses to stimuli. Cool. Next, define the term differentiation, dedifferentiation, redifferentiation. I already told you, just go through it. So, where the meristematic cells become specialized for specific function, as in they have a proper function now. Meristematic cells used to divide, but now they have stopped dividing, they have a proper function. This is known as differentiation. It can happen with any cell. Now, dedifferentiation, when it again regains its ability to divide. Okay, it does not have a proper function, it is just dividing itself. Now, redifferentiation, when it stops the division and says that I have a proper function as of now. Okay, now mention the source from which the following growth hormones were first isolated. Auxin, my dear children, it was isolated from oat coleoptile tips. Now, this is something that you have to by heart. I cannot do anything about it. Kinetin, it is discovered from herring sperm DNA extract. Gibberellin, it is isolated from Gibberella fujikuroi fungus. It is a fungus. Okay, so you have to remember oat coleoptile, herring sperm DNA and Gibberella fujikuroi. Yes. Now, both growth and differentiation in higher plants are open. What do you mean by that? Open as in growth in plants is open because the cells continuously divide. It will obviously happen always. That is why it is open. Why is differentiation open? Differentiation as in at any point of time the cell will be differentiated. Any point of time they can again start off division. So differentiation is open because cells can change their function even after specialization. That is needed for them, right? They cannot move around, so obviously they should be having that ability. Now last, describe arithmetic growth, geometric growth, absolute growth rate and relative growth rate. Arithmetic growth as in growth at a constant rate producing a linear curve. So where the curve goes like this. There's a linear curve that is there where you say that the growth rate is what, my dear children? Constant. This is known as arithmetic growth. For example, if you talk about root elongation, it will definitely happen like that. It will grow on, grow on, grow on, grow on, grow on. That is kind of rare, but it happens in there. Now, geometric growth is basically when sigmoid curve happens. Okay, so there's a lag phase and then there's an exponential phase and then there is a stationary phase over there, because the resources are limiting over there. Okay, so growth occurs exponentially, forming a sigmoid curve, cell division in meristems. So in meristems they divide and later they get specialized. So this is what geometric growth, my dear children. Now, what do you mean by absolute growth rate? So measurement and the comparison of the total growth rate per unit time is called as absolute growth rate. You have to remember this particular definition. It's there from your NCERT itself. I cannot do anything, but I can explain you what does that mean. So measurement and the comparison of total growth per unit time is called absolute growth rate. So suppose this particular leaf A had 5 centimeter, it was 5 centimeter square, now it is 10 centimeter square. So if you want to find the absolute growth rate, what do you do? 10 minus 5 you do, that is the absolute growth rate is 5 centimeter square. Measurement in the comparison of the total growth per unit time. Per unit time, what is the total growth? Now relative growth rate is what? The growth of a given system per unit time expressed on a common basis, that is per unit initial parameter. That is kind of very complex. I'll just tell you. I'll just tell you how it is calculated. So absolute for this is this 5. Similarly for here also 55 minus 50, it is what? Again 5 centimeter square. If you want to find the relative, what do? You put absolute growth and you also calculate the initial one. So what is the initial? So initial growth into 100, that is what relative growth rate. So absolute growth rate over here was 5, the initial was also 5, into 100 if I do, my dear children, what do I get? Oh yeah, sorry, oh my god, just a second, I did a mistake, yes, there was no mistake. Okay, so basically if I do, then it is what? 20, right? 20 into 5, my dear children, again it is what? Am I doing it correct? Yes, it is 100. That is a relative growth rate. But over here, let's calculate that. Over here, it was what? Absolute growth rate is 5, but the initial was 50 and then it is 100. So here it is 10 and here it is 100, my dear children. That's the difference that you get. So this is the relative growth rate. For relative growth rate, over here it's 100, there it is 10. I hope you got it. Okay, I thought I forgot the formula. Yes. Now, chemical coordination and integration. So this chapter has only 5 marker question to offer you as of now, what we think. Okay. So previous year it was 8 marks question that you used to get from here. Okay. List, that is total number. List the functions of thyroid hormone. So these are the first 5 marker question. So thyroid hormones plays an important role in what, my dear children? Metabolic rate. Always remember thyroid hormone is for metabolism of your carbohydrates, fats, lipids, whatever you have. Okay. So these hormones support the process of red blood cell formation as well, RBC formation. They help in metabolism. They help in maintenance of water and electrolyte balance as well. And they also secrete a protein hormone that is known as thyrocalcitonin, which regulates our blood calcium levels as well. So these are the things that you should definitely be writing in the paper if you're asked about thyroid hormone. Going ahead, list the hormones secreted by the following. Hypothalamus. So hypothalamus has thyrotropin releasing hormone. So what I will tell you a just small thing. Okay, hypothalamus tells the pituitary what has to be released or what has to be inhibited. Pituitary will now tell other glands what has to be released and what should not be released. Okay. Now hypothalamus, it releases thyrotropin releasing hormone. So thyrotropin releasing hormone will go act on pituitary gland. Okay, and will tell the pituitary to release thyroid stimulating hormone. Now this thyroid stimulating hormone will actually go to the thyroid gland and will make it release thyroid hormone. So that's how it works. Gonadotropin releasing hormone, as in you're talking about FSH and LH. Okay, so this goes and acts on pituitary. Now the pituitary will be releasing FSH and LH. Now growth hormone releasing factor, now this will be hormone, this will be acting on the pituitary and it will directly, so pituitary gland releases directly FSH and LH and also growth hormone. Now corticotropin releasing hormone, my dear children, it will stimulate ACTH, that is adenocorticotropin hormones, my dear children, which is released by adrenal glands. So this will release, this will go act on pituitary, the pituitary will be releasing this, which will go and act on adrenal glands, and that's how things will be working. So you should know about this thing. Okay, this is how things work. Now they can be releasing inhibiting hormones. For example, thyrotropin inhibiting hormone. In that particular case, it will go and act on the pituitary and it would not allow basically TSH secretion to be happening, or basically thyroid hormone formation to be taking place. Now gastrointestinal tract, over here they have gastrin, secretin, cholecystokinin, and what do you have? You have gastric inhibitory peptide as well. So I have written the functions of each, you can just go through. Okay, so gastrin basically goes and acts on gastric glands and tell that you have to secrete what? Hydrochloric acid, HCl, which is needed for breakdown of all the unwanted things and also for activation of an enzyme, that is pepsinogen. Secretin, it acts on the exocrine pancreas, my dear children, and stimulates secretion of water and bicarbonate ions. Bicarbonate ion really much important for all the other pancreatic enzymes to act. Now, cholecystokinin basically acts on both pancreas as well as gallbladder, stimulates the secretion of pancreatic enzymes and as well as the bile juice, which is being released by the, which is released by the liver but stored in the gallbladder. Now, gastric inhibitory peptide inhibits gastric secretion and also motility. So all these functions you should be knowing. list the hormone. Okay, now thymus, you have thymus secretes one hormone, and that is thymosin, my dear children. Thymosin plays a major role in differentiation of T lymphocytes. T lymphocytes basically get differentiated in the thymus, so obviously you need thymosins. Pituitary gland, what do? You have anterior and posterior pituitary. Anterior pituitary produces its own hormone, that is growth hormone, my dear children. Also prolactin, thyroid stimulating hormone which will act on the thyroid gland, adrenocorticotrophin hormone which will act on what? Adrenal gland. Luteinizing hormone is a own hormone, and follicle stimulating hormone is also its own hormone. Now you have melanocyte stimulating hormone which will go and act on melanocyte as well. Posterior pituitary stores and releases hypothalamic hormone, that is oxytocin and vasopressin. So they don't have their own hormone. You get oxytocin and vasopressin from the hypothalamus. It is being stored in the posterior pituitary and it being released from posterior pituitary as well. Adrenal releases adrenaline and noradrenaline, cortisol and aldosterone as well. Works. And also the sex corticoids, okay, and also the sex corticoids as well. Now write one physiological function of LH, GH, oxytocin, vasopressin and TSH. LH, luteinizing hormone, my dear children, it is a one. In the case of females, it will lead to ovulation. It will lead to ovulation, what's a release of egg. Whereas in the case of male, it will be helping in testosterone production. That is androgen, okay, it's a male hormone. Growth hormone, what about that? It helps in body growth and cell regeneration. Oxytocin, what about that? It induces labor contraction. During the labor, during the expulsion of the baby outside, you need oxytocin. Vasopressin, my dear children, you need, it is an antidiuretic hormone, controls water retention in the kidneys. So if your body is losing a lot of water, that will be a problem if you do not have it enough. So basically, vasopressin is the one that allows to take up that water, reabsorb that water. Now TSH, I told you, it's thyroid stimulating hormone, it regulates the thyroid hormone secretion. Going ahead, a sample of urine was diagnosed to contain a high content of glucose and ketone bodies in the urine. Okay, now based on the observation, write the following. Which endocrine gland and hormone is related to this condition? My dear children, pancreas is the one. Whenever you find glucose, just understand you're talking about pancreas. Okay, if you have high content of glucose over there, probably it's because of insulin. So because insulin might not be working over there. Okay, there's no insulin working over there. So this disease is caused by pancreas. This is caused by because of the gland, that is pancreas, and the hormone that is responsible for this related condition is what? Insulin. It is secreted from the beta cells of the pancreas. Now name the cells on which the hormone acts. So it will act on liver cell, it will act on muscle cells and also adipose tissue cell, and tell them that you have to convert all the glucose. Usually insulin tells that you have to convert all the glucose into glycogen and store it over there. Okay, that's how things work. Now, what is the condition called? This is known as diabetes mellitus, my dear children. And how can it be treated? It can be treated by giving insulin therapy. Then that's the only way it can be treated. Cool? Chalo. Now, again, these are the 5-marker questions that you should definitely not miss. Schematically represent the action of estrogen. Name the endocrine gland in which the following hormones are synthesized. Write one function: melatonin, glucagon, thyrocalcitonin, progesterone, cortisol. All of it you should be knowing. What are gonadotrophins? Enumerate their action in males and females. You're talking about LH and FHS. They are known as gonadotrophins. Okay. Write a note on the pineal gland. Catecholamines are hormones of fight or flight. Justify. So adrenaline and non-adrenaline are coming under this category. Okay. Enumerate the role of ovarian hormones in the female: estrogen, progesterone, things like that. Glucagon and insulin are antagonistic in their function. Why? Because glucagon says that, hey, glycogen, let's break down into glucose. Whereas insulin does what? The opposite. It takes all the glucose and converts into glycogen. So you should be knowing about all this thing. Locomotion and movements, my dear children. Now this is a chapter that gave 7 marks the previous year, and here are certain 5-marker questions that I have put. I think I have explained like this. Now, okay, wait, yeah. So what is a skeletal system? Write its 2 divisions. So skeletal system, it provides support, movement, protection and everything, my dear children. It consists of bones and cartilage. It is divided into 2: axial skeleton as well as appendicular skeleton. Axial consists of skull, your vertebral column, your ribs as well as your cranium, everything that provides protection. You have almost 80 bones over there. You have to explain about each of them in a detailed manner if it's of a 5-marker question. Okay, in vertebral column, how many things are there, in skull, how many bones are there, everything you should be knowing about it. Appendicular skeleton, 126 bones. It is basically including the limbs, that is forelimb and hind limb, and also the girdle, that is pectoral and pelvic. Talk more about it because this is not enough for 5 marker. Now I am just giving you just what you should be including. Okay. Now enlist the bones forming the forelimb. So over here we have the humerus, then we have the radius and the ulna, and then we have the carpals, then we have the metacarpals, and then we have the phalanges over here. So humerus is the upper arm bone, radius and ulna bones are the forearm, carpals is the wrist bone that we have 8 in number, metacarpals we have 5, that is palm bones that you can feel over here, and phalanges is the one that is over here. Okay, they are 14 in number, they are the finger bones. Now name some skeletal system disorders. So osteoporosis, my dear children, where there is decreased bones density. Why? Because there is calcium deficiency. This happens to people who have thyroid hormone problem. Arthritis, inflammation of joints leading to pain and stiffness. Gout is basically when uric acid crystals comes in between the joints. Now rickets, when vitamin D deficiency happens. Okay. Now next, what are the types of joints and the location in the human body? A joint is what? A junction between two bones, my dear children. It can be a fibrous joint which is not movable. For example, in the skull, we have fibrous joints. Okay, they are not movable. Now talking about cartilaginous joints, in between these joints, what do we have? You have cartilage, which is why you can at least do some amount of movement, limited movements. For example, intervertebral disc, in between our vertebrae, you have this thing. Synovial joints is where you have a fluid, that is synovial fluid between two joints. Okay, there is a ball and socket joint, hinge joint, pivot joint, gliding joint, saddle joint. All of them, they come under this particular category. Okay, ball and socket over here in the shoulder and the hip. Okay, hinge joint in the knee and the elbow, pivot joint between the atlas and the axis, which is there over here, and then you have gliding joint in between the wrist and the ankle, saddle joint between the thumb. Okay, which is why you are able to move like this and this. That's all. Going ahead, these are the 5 marker questions that you should not be missing out. Describe the structure of actin and myosin. Explain the steps involved in sliding mechanism of muscle contraction. Describe the bones of the skull in man and skeletal and cardiac muscle. Sliding filament theory. Sketch the diagrammatic view of human skull. Again, a diagram which you should not be missing. D. Okay, cell cycle and division. From here, my dear children, I won't be explaining anything. From here, the 6 marker thing starts. You have to just take a screenshot of this because it's already 50 minutes. I don't want to go ahead. It might be too boring for you. So please take down these questions. For 2 marker, you have to prepare from cell cycle and cell division. Okay, for 3 marker, you get these questions over here. Mitosis, meiosis difference is very important. If you know about all the stages of mitosis and meiosis, you might be able to basically write all the questions over here. Again, you have a neatly labeled diagram of cell cycle. So you should be knowing about this thing. Okay. Again, respiration and plants, 5 marker questions are these. Schematic representation of glycolysis, something very famous, something that usually comes in. Okay, Krebs cycle, citric acid cycle, TCA cycle, anything, basically the same thing that could come now in with any name. Now, discuss the respiratory pathway in an amphibolic pathway. Is an amphibolic pathway. Difference between aerobic and anaerobic respiration, what happens in mitochondria, what does not happen. Note on fermentation, either way, all for 5 marker. Biomolecules, again a 6 marker chapter. Okay, write a note on cofactor, enzyme classification, mechanism of enzyme action, classification of proteins, function of proteins, list any 5 functions of proteins with examples. Okay, most abundant, what's say, you have to talk about all this thing. You should be knowing about all this thing over here. Okay, so just go through your NCERT, you will be able to do it. And let me know if you're not finding the answers, I'll help you out. Okay. Now, question is an exothermic or endothermic reaction. That is this thing. I have solved in the model paper question paper. This is a model paper question. So, please go and watch it if you do not get the answers for this. Considering E for enzyme and S for substrate, depict the nature of enzyme action. Explain the 4 steps of catalytic action of enzyme. Lipid are not strictly macromolecule, substantiate. Again, this also I have discussed. Deuterion, very important. Metabolites, factors affecting. Okay, all these things, read. It's 5 marker. Okay, just go through your NCERT. At least you know this. So what do? You take the PDF. If you are not sure about anything, you open the PDF of your chapter, whatever is there. Okay, and then Control F. You look for the terms. For example, you put macromolecule in that chapter. Wherever there is macromolecule, you get that. Read that paragraph. Okay. Now going ahead. Body fluids and circulation, again 6 marks, my dear children. You should be able to draw this. This is a diagram. Draw a standard ECG and explain the different segments of it. Very much important. So if you learn about ECG, a lot of questions will be solved. Now apart from that, again there's a diagram of human heart. What is cardiac cycle? Explain the stages. The birds have double circulation as requirement for flight is substantiate. Write a note on condition and symptoms of erythroblastosis fetalis. Okay, when the bloods basically mix. So all this thing you should be knowing. Again 5 marker things. Breathing and exchange of gases, these are 2-marker questions. Okay, give the graphical representation of oxygen dissociation curve, very important. What is tidal volume? I already explained in the PU1 model paper. Expiration and inspiration, residual capacity and functional residual capacity, vital capacity and total lung capacity, everything is given together. Very easy for you to understand, just go through it. Okay, I am telling you it's easy, that means it's easy, and whenever you have a question and you are just finding the answer, it becomes all the more easy for you to recollect things. Cool? Now, photosynthesis in higher plants, my dear children, what are special anatomical features displayed by leaves of C4 plants and Calvin pathway, differentiate between C3, C4 plants, explain the Blackman's law of limiting factor, all of them 5-marker. I hope you're taking the screenshot. Okay. Schematically represent non-cyclic photofossilization, important. Diagram. Okay. Now, plant kingdom. So over here, salient features of pteridophytes, gymnosperm, mosses and algae. I have discussed at least a bit of it. Okay, economic importance of algae. Write a note on cell wall, flagella number, food storage of different classes of algae, red algae and brown algae. Okay, differentiate between I think it was red and brown. Okay, explain the terms protonema, antheridium, sporophyll, isogamy, archegonium. Pteridophytes exhibit heterospory. Explain why, because they have 2 types of spores. That is what you mean by heterospory. So just go through it. Okay, plant kingdom. Excretory products and their elimination, again 6 markers. So over here, define the terms glycosuria, ketonuria, diagram is there, nephron. Okay, very much important. Again, I have told you in PU1 model paper, I have explained it, I have drawn it by myself. Skin also helps in elimination of excretory waste. Substantiate, describe any 3 disorders of excretory system, counter current mechanism, very much important. Okay, go through it. Morphology of flowering plants, these are the questions. Be sure about the placentation, be sure about aestivation and things like that. Okay. Diagram is there. Now there is a question. Yeah, I have solved this in PU1 model paper. Okay. Neural control and coordination. These are the questions that you should be going through. 3 marker. Okay. 3 marker, they can like come in a 5-marker question as well. We never know. So be prepared. Biological classification, it was out of 4. Okay, 4 marks were in the previous year. So this time board is not organizing, so we cannot be sure of anything. But yes, if you cover these topics, it will be really important, easy for you to do. Okay, and anatomy of flowering plants. Again, you have a diagram over here. All these things. Okay, I have discussed this and a bit of this in the PU1 model paper. Okay. Structural organization animals, frogs exhibit sexual dimorphism. Frogs can live both on land. Again, I have explained this. Okay. List any 4 sense organs. Frogs are beneficial. Parts of excretory system. List the types of endocrine found in the frog. Differentiate between blood and the lymph in the frog. So why I am saying that I have explained is that if you do not know the answer, you can go search there. Okay. So this is 2 marker questions, my dear children, which have come. Now living world, which is the last one. So here we have what is binomial nomenclature, all these things, and rules of binomial nomenclature from here to here. So that's all, my dear PU1 aspirants. I hope that this particular video would have been beneficial for you. Do let me know if there are certain questions that you are not able to solve. So what do? Write down the question and say that, ma'am, can you please explain this? Okay, and I will be surely making a video on it. So do not think that I won't do that. It's for you that we are doing this. Your exams are coming up. A lot of you might have started also. So please do let me know that. And if it helped, do not forget to hit the like button and also the subscribe button to our little channel. That's all. Bye-bye. Take care.